All right, today we're diving into something massive, something that's literally changing the world as we speak. We're talking about genomics, the science of decoding the complete instruction manual for life itself. Let's get into it.
Just think about that for a second. Really, what if you had the complete blueprint? Every single part, every instruction that makes a living thing, well, live. That's not science fiction. That's the incredible world we're exploring today.
Okay, so let's get our terms straight. What exactly is genomics? It's not just about looking at one gene here or there. No, this is the big picture. It's the study of the entire set of genes, the whole shebang, how it's all put together, what it all does, and even how it changes over time.
Now, to really wrap our heads around this blueprint, scientists have to look at it from two different angles. They're totally connected, though. Think of them as two sides of the very same coin.
So, on one side, you've got structural genomics. This is all about making the map, the complete parts list. It's asking, what's in here? Then, on the other side, you have functional genomics. This is where it gets really cool. It asks, okay, what do all these parts actually do? It's the difference between having the blueprint of a car and actually understanding how the engine works, how the electronics talk to each other, and what makes it go.
So, how in the world do you even start to map something as ridiculously huge and complicated as a genome? Let's tackle that first big challenge, just creating the map.
Well, think about it like this. If you're mapping a vast, uncharted continent, what do you need first? You need landmarks, right? Same idea in genomics. Scientists use what are called molecular markers. These are just unique little snippets of DNA that stand out, acting like signposts, helping them find their way through billions and billions of genetic letters to locate a specific gene. They are absolutely essential for navigating this thing.
And this is where it gets really interesting because there are actually two different kinds of maps. A genetic map is kind of like a subway map. It shows you the order of the stops, gene A comes before gene B, but not the exact distance. A physical map, on the other hand, is like a super detailed satellite map. It gives you the precise physical street address, measured right down to the exact number of DNA base pairs. You really need both to see the whole picture.
Okay, so you've got a reliable map, you know the lay of the land. Now comes the next monumental task, to actually read the entire book of life, letter by letter. So how's that even possible?
Structural genomics really boils down to these three key steps. First, you build those super detailed maps we just talked about. Then, step 2, you sequence the whole thing. That's the process of reading every single A, T, C, and G. And finally, step 3, you figure out all the proteins that the genome codes for, because proteins are the tiny machines that actually do all the work in our cells.
All right, let's pause. We have the map, we've read the sequence, we basically have the complete parts list for a living thing. But what does it all do? This is where that static blueprint starts to come alive.
And that brings us to functional genomics. This is all about the why and the how. If structural genomics asks what's there, functional genomics comes in and asks, okay, but what does it do? How do these parts work together? When are they turned on or off? This right here is the fundamental question that drives this entire side of the field. It's one thing to have the raw code, but it's a completely different challenge to understand the language it's written in.
And what's so crucial here is that functional genomics helps us answer the really big questions. It helps us figure out which genes are turned on in a brain cell versus a skin cell, for example. How active are they? What's controlling them? And maybe most importantly, it helps us see how this whole beautifully complex system changes when something goes wrong, like in a disease.
Now, you can probably imagine, to answer questions this complicated, you need some seriously powerful tools. So let's take a look at the high-tech toolkit scientists have at their disposal. First up is a total game changer, next-generation sequencing, or NGS. This technology lets scientists read millions and millions of DNA fragments all at the same time. It's what took genomics from a decade-long billion-dollar project to something that can be done in days for a fraction of the cost. It's just unbelievably fast and powerful.
And this isn't some far-off concept. These are the real-world workhorses, the platforms from companies like Illumina and others that have truly powered this entire revolution. Next up, we've got mass spectrometry. Now, if NGS reads the genetic instructions, mass spec identifies the workers, the proteins. You can think of it as an unbelievably precise scale for molecules. It lets scientists see exactly which proteins are on the job in a cell at any given moment.
And check this out, DNA chips, also known as microarrays.
These things are basically tiny, miniature laboratories on a little glass slide. They allow researchers to take a snapshot of a cell and see which of thousands of genes are turned on or off all at once. It's just incredible.
So we have the structure, and we have the tools to understand the function. What happens when you put it all together? That's when you get a truly powerful, complete picture of life.
This is what's known as a systems biology approach. By combining all the information from these incredible tools, scientists can finally stop looking at just one piece of the puzzle. Instead, they can see the whole system, the whole living network, all at once, creating a complete line of sight from a single gene all the way up to a biological function.
And this quote really just says it all, doesn't it? The whole point, the ultimate goal, is to get this complete picture of how our genes make us who we are, in sickness and in health. This is where the real breakthroughs in medicine are happening right now.
And that leaves us with one final, pretty profound question. For the first time in human history, we can read the blueprint of life itself. The question for all of us now is, with this incredible, almost unbelievable power, what should we build next?